Viruses are the most common biological entities on our planet, infecting virtually all cellular organisms. While viruses were originally believed to be smaller than 200 nanometer in size, the discovery of the first giant virus, Amoeba polyphaga with a genome and particle size comparable to bacteria has changed our perception of the viral world and the complexity of viral particles and their genomes. All known giant viruses belong to the Nucleocytoviricota phylum, formerly known as nucleocytoplasmic large DNA viruses, NCLDV, and have double-stranded DNA genomes. Giant viruses Nucleocytoviricota are significant lethality agents of various eukaryotic hosts. Although metagenomics indicates their ubiquitous distribution, available giant virus isolates are restricted to a very small number of protist and algal hosts. A new, unusual giant virus has been discovered at a wastewater treatment plant near Vienna. The University of Vienna researchers have discovered viruses named Naegleriavirus that infect the dangerous Naegleria fowleri, offering new insights into virus biology and potential water treatment solutions. The single-celled organism Naegleria fowleri ranks among the deadliest human parasites. Researchers at the University of Vienna's Center for Microbiology and Environmental Systems Science have identified viruses that target this dangerous organism. Named Naegleriavirus, these belong to the giant viruses, a group known for their unusually large particles and complex genomes. The team details their findings in the prestigious journal Nature Communications. Naegleri species are single-celled amoebae found globally in water bodies. Notably, one species, Naegleria fowleri, thrives in warm waters above 30 degrees Celsius and causes primary amoebic meningoencephalitis (PAM), a rare but almost invariably fatal brain infection. Researchers from the University of Vienna Center for Microbiology and Environmental Systems Science (CEMES) has now isolated giant viruses that infect various Naegleria species. Giant viruses, scientifically termed Nucleocytoviricota, are a virus group identified just 2 decades ago, primarily infecting single-celled organisms. These viruses rival bacteria in size, boasting unique structures and genetic traits previously thought exclusive to cellular life. Their discovery sparked debates over the definition of viruses and the origins of life. The newly discovered Neoria viruses were isolated from a wastewater treatment plant in Klosterneuburg near Vienna and represent only the 4th isolate from a group called Klosterneuviruses. This discovery and the characterization of Neoria viruses were made possible through international collaboration with researchers from the universities in Poitiers, the Canary Islands, and the US-based Joint Genome Institute. Neoria viruses are taken up mistakenly as a food source, but destroy their amoeba hosts within just few hours. They exhibit a structure familiar in giant viruses, infecting host cells via a so-called Stargate structure that facilitates DNA entry. Within hours, a structure known as a virus factory forms inside the amoeba cell, replicating viral genetic material outside the nucleus and assembling hundreds of new virus particles to keep the host cell alive during this process. Naegleria likely use special proteins that suppress the cell's natural immune response, preventing premature cell death. Only after successful viral replication does cell destruction and virus release occur. Viruses are employed in phage therapy to combat bacterial pathogens. The newly identified Neoria viruses may not be suitable to treat Neoria infections, given the challenging accessibility of the brain where infections occur. However, this discovery opens the door to the possibility of preventative treatment of at-risk water bodies, such as during swimming pool water treatment, but this would first require further research. Regardless, the discovery of these viruses will enhance our understanding of both Naegleria biology and the viruses that infect them. The Naegleria infecting virus isolate reported here could represent a first step towards a viral biocontrol agent for Neoria fowleri. Importantly, it allows scientists to study processes like programmed cell death and antiviral defense in Naegleria fowleri, which will ultimately contribute to a deeper understanding of the biology of this serious human pathogen. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.